All right, and we are live. I am live. Good morning, everybody, and welcome to our Science Saturday. It is a beautiful day, and we are down at the University of the Virgin Islands Marine and Science Center, and today we are learning all about corals and how they grow and really interesting things about their shapes and fun stuff like that. So we are very lucky to have your favorite coral scientist with us today, the one and only, you know him. Good morning, Joe.
Morning, everyone. I gotta say, Kitty, I'm pretty excited because we've been doing a lot of Science Saturdays, but this is the first one in a while that we've done on coral. So, the ones I know and love the most. And so what Kitty and I were talking about was, one of the big questions that we get a lot when it comes to corals and, how they build these incredible reef structures, these coral reefs that we value so much, is, how exactly they're growing and all the weird and interesting shapes that they grow, right?
Yeah, I want to know.
Well, so for example, these are a couple of Kitty and my personal favorites. This is a skeleton of what I believe is a maze coral. It might actually be a different coral, but it looks like to be like a small maze coral here. And you can see it's just, in addition to kind of growing out and creating that boulder structure that we know reefs to have, you notice it has all these little intricate substructures, right? These are called septal teeth, or septa. They're kind of growing all along here, and it creates this really complex structure. You can see it again. This is a different coral. This is a this smooth flower coral, I believe. Yeah, Eusimilia fastigiata.
And how do we remember the name of that one?
This one?
Yeah.
Eusimilia fastigiata. I don't know, if you have a good way of remembering that entire Latin name really easily, I'd be happy to hear it.
Well, luckily, I have a very good friend who is also a coral nerd, and she taught me that you smelly like a flower, because when that is alive, each polyp looks like a flower, and so together it looks like a bouquet of flowers. Right. So you smelly like a flower.
Right. So I think this is really interesting, right? We have corals that are growing in all these weird, crazy, complex shapes. And the cool thing about nature is that's not an accident. This is very intentional, and there's a reason behind all of this that's going on, right? So
I assume so, yeah.
So let's back up a little bit. Corals, remember how they get their food? We've talked about this in other Science Saturdays, so I'm seeing if anyone remembers.
How do corals get their food? Do we remember? I don't.
You don't? So,
no.
A quick refresher. Corals actually have special partner symbionts. So these are little tiny cells that live within the coral cells. They're called photosymbionts. And that's because they can actually take in sunlight, like plants do, and make energy and food for the coral. So really the way that corals get their energy is the more sunlight that they have. And that's one reason that they have to grow in this crystal clear tropical water that we have here in the VI, is they need that light to come through that water and feed the symbionts, so feed those little cells that are living inside and provide food for the coral. And so this actually, believe it or not, ties into exactly why we have so much complex structure on our corals. Believe it or not, because it's not just the symbionts that need that light and need that food that's produced from it. It's in the coral's best interest to also make sure that symbionts get as much light as possible. And so it's really interesting when you think about it. Let's say you've got like a flat plate and you've got some symbionts spread across, kind of like chicken pox, right? And you shine some light down onto it. Well, those symbionts, where they're sitting in a flat area, they're going to capture that sunlight, but some of it's going to go in between, where there's not symbionts, right? Going to pass through. And so what that light does is when it passes through that tissue where the symbionts are living, it may not be absorbed by all the symbionts. And so with the white skeleton of the corals, that light is all bounced right back up. And so that means the symbionts, not only are they getting light from above, from where the sun's coming from, but they're also getting the reflection that's coming back up.
Ooh.
And let's say,
Smart.
now you've got two chances to get that light. Well, let's say you still miss it. Let's say you're a symbiont, you're a little bit small, it just wasn't quite the right angle, right? Well, this is where this kind of complex structure comes in. Rather than bouncing this back up like a flat plate, all of the structure here allows it to bounce all around in all kinds of angles. And so for every beam of light that comes into here, you're going to have that beam of light bounce around, 10, 20, 30, hundreds of times, giving every single symbiont a chance to get all of that sunlight and maximize that light. Does that make sense?
That does make sense. That's fascinating.
Well, so I thought a really easy way to demonstrate that would actually be a handy dandy laser pointer that we have here. This is a scuba diving laser pointer. And so you can see, the cool thing about laser pointers is, right, so this is light here. But the cool thing about laser pointers is it focuses the light into one area, and you can see there's not really any bleed around. Like there's a little bit of reflection. If I put my finger like that, you can see a little bit of light is coming up, but generally speaking, it's all kind of going right to one spot. So now let's see what happens when we shine one tiny little beam, because normally the corals are used to getting, big, wide range of like sunlight that's all around us. And he's getting like a single beam of light. But let's see what happens when we shine the light onto the coral. So right now we've got that focus beam here. And now when I shine it inside here, what do you see?
Ooh, oh, I can totally see how it's bouncing around and no longer one solid stream of light.
Right, so you're seeing that there's kind of reddish all over the coral, right? There's like light that's appearing all in everything. And that's because of these septal teeth here, the angle that's down here in the bottom in the valleys. Because of all of that, this light, even no matter how focused, no matter how much one direction it's going, when it strikes the coral, it immediately bounces all over the place and you have light going all over and everywhere else. And sure, some is reflected back up. That's how we're able to see it, right? If it wasn't being reflected, it would just look like it's dark there. But that reflection also gets caught, remember, in the tissue that's on top of the skeleton where those symbionts are living. And so this allows corals to take in just a little bit of light and use almost all of it, which is really incredible for the corals themselves. And that's what allows these corals to grow and where there's a lot of light and not necessarily like a lot of food in the water. And you see here, and just to show, so here's another one here. This is the You Smell You Like a Flower. And you can see the same thing. When I shine it right down in the middle of that polyp there, it immediately, you see red all over the skeleton. You can see it, and if you look really close, I don't know if we can see it on the camera, but it looks like there's just like a bunch of little particles, just kind of like, it almost looks like static on a TV in the skeleton.
Oh, yeah.
Yeah, I hope you guys can see that.
I hope so too. Putting the iPhone to the test.
Right.
If you can't, let's start a fund to get me a fancier iPhone.
Aha.
In the name of science.
Of course, in the name of Science Saturday. So, this is kind of one of the, so you can kind of use this as an understanding. This is one of the main reasons that corals grow and the interesting different shapes they do. The big question about, what's kind of shaping these corals is where can they get the most amount of light? And that's what allows them to grow in places that you would never expect corals to grow necessarily. As long as they can get enough light, they can grow there.
All right,
Right?
makes sense.
Yeah.
Yeah. As long as we're happy enough, we can grow there. Enough food to stay
and even,
satiated.
Right, and this is the reason that also that we have corals growing in different shapes and different sizes as well. Because some corals have figured out better ways to do this in different environments. So we think about like in shallow water, right? Can you think of some of the corals we see in like shallow water?
Yeah, I see acroporas in shallow water.
Like elkhorn and staghorn coral, right? These really big species, right? They're really big and everything. So acroporids or any of these elkhorn and staghorn are actually really incredible corals because they're some of the fastest growing in the entire coral community. And so they grow in this really shallow water where there's tons of light, right? And in fact, there's so much light that if the light misses the coral and bounces off the ground somewhere else, that's enough light for the coral to grow. So it has coral tissue right facing the sun, it has coral tissue in the shade that's getting enough light, and because it's able to absorb all of that light so efficiently, it's able to grow twice as fast as some of the other coral species that we have.
And that's why they're so good for coral restoration,
Right.
which we'll do another day.
Right. And so this is one reason that scientists and conservationists are working so hard to restore these areas is once we outplant them on the reef, they're going to grow really quickly. They're going to create a lot of really strong structure really quickly. And we used to have a lot of it. Back in the 1970s and 1980s, we first started seeing a major die-off of these species. But it used to be that along the barrier reef, and I'm sure some people will remember this, along the barrier reef, you would just see tons of elkhorn and staghorn. And that's really where our big like reef break, that wave break energy was, right? Because these corals were so good at absorbing tons and tons of light, they were able to create this huge structure and break the waves for us.
Interesting.
Right? And so now, why do you think that things like the elkhorn and staghorn only grow in the shallow water and then don't grow in the deeper water?
I'm guessing something to do with light and their ability to absorb it.
Right. So these corals are so well adapted to the highlight that they don't necessarily do well when you go deeper and deeper in that water and there's less light present, especially because they're so used to light being all around them. And so this is actually another adaptation that you see for shallow water corals. This Eusomilia is showing a good example of it, but I can get some other corals out here. These are all coral skeletons that were collected as a part of coral disease work. So these were all infected corals that were going to die, and so we brought them back to the university in an attempt to kind of treat them and save them. But unfortunately, these corals weren't able to make it. But we can use them here as a demonstration for instead of bringing real live corals out of the water, which we don't necessarily want to do. So one thing that's interesting that you see in other shallow water corals, these brain corals, is you see how they're kind of round, right? What we call that like semi-hemispheric. So this is an example of one, and then this is a grooved brain coral, Diploia labyrinthiformis. This is an older sample, but you can see how it's very round, right? And that's because even if this coral, so we can tell the coral was probably, mounted right here on the reef, and this is kind of, you can see where it probably grew, starting here, growing out and up all at the same time, just kind of making a dome. And that's because as this piece is growing continuously upward, so this is where this tissue is here can align, or this skeleton here, as kind of generally one polyp moving upwards, right? And then you can think of it cloning itself, and that's how it gets wider and wider. So this coral is going through both vertical growth and horizontal growth at the same time, kind of at the same rate, making this dome, right? And that's because this tissue is getting enough light to where it can replicate itself and it can go, more horizontal, but it's also getting enough light that it can create this rock, this calcium carbonate structure, and grow upwards. So it makes this dome shape. And these guys continue to grow outward like this as well, because even though they're not getting the direct sunlight down, there's so much sunlight in the shallow water that this side light is more than enough for them to grow outward. And this is kind of what decides how these corals grow in the shapes that they grow. So can you imagine, if we're in the shallow water, where we've got all this light, there's light coming all around, you can see it's bouncing, there's not a lot of shadow here, even though the sun's like way up there. But then if we move into lower light, so now the light that's over here, you can see that it's a lot darker on this side compared to kind of up here. This is the lower light. This is what it's like on a deeper reef, for example. And so in this area, you're really light limited. You have to use the light that you have that's coming straight down. That light that's bouncing up and bouncing around on the reef isn't enough. And you really have to maximize that structure that maximizes the light from the top. And this is why when you go to these deep offshore reefs or any reef in a deeper water, what you're going to see is the corals will go from being these big mounds and these bulbs like this to being more flat plates and spreading out because they're just trying to maximize that light. Almost like I always think like a satellite dish. So then comes the question, why do we have all kinds of crazy different shapes of corals all over the reef, right?
Why do we? I think about that all the time. It's a fascinating question.
Do you have a favorite shape?
A favorite coral shape.
Or have you ever found a coral that was like your favorite shape?
So I really love the Eusomilia. I think that they are really interesting and they don't get very big, and so they're kind of like little gems in the reef. However, I have seen pictures, and I've never been diving there, of some of the deeper water plate corals. And I think that is beautiful, and how they kind of grow over each other. And you can see the competition for space and so many different shapes all working together to create that reef.
Right. And so this is a really good point. So these corals, when it comes to where they're settling, they have to choose where they're going to settle, where they can grow the most, and they can keep getting the food that they need and things like that. And so that competition over space, like you were saying, is a really big deal for corals. Wherever you settle, you need to grow out from that, right? And so when we're talking about a reef, you can think of a reef area as kind of an area, a certain amount of area where coral can live. And that's a finite area. There's only so much that's present. So you can think of this maybe as a miniature reef, right? And if you look over the top here, you could think of this as the finite area that you can grow in, right? So, and this is how we talk about how corals create structure and everything.
Sorry guys, I'm short. I hope I'm getting this lined up right.
I can move it over
We're
here
good.
a little bit. So,
This is the reef,
right.
the ocean.
So we have the reef here, and so this is this is how we can tell that corals create this structure and things like that. This is just kind of a way to demonstrate that. We have 2 kind of buckets of water here. This is going to be our big reef that we'll eventually build. But just to show you guys how we're going to measure the differences, what we can do is we can do a volume displacement experiment where we look at how much water is displaced by these colonies. What that tells us is the volume of the coral, how much space the coral is taking up, right?
This is real science, guys. Volume and coral.
So admittedly, we are on a bit of a tilt here, but what we can do, just for now, we can make a little mark here, right on the water line. We'll make one down over here too. And now, when we add our corals, so we know that our water line is here when we don't have any corals in there. So let's add our corals in here. So there's one. Just go ahead and add this one. He can't get all the way in the water. Now, what you see is interesting. Let's see. Let me see if I can get one more tiny coral here. I don't really have it. Oh. No, I did. Oh well. Yeah. That one fit. He might have to wait until our big reef.
Maybe.
Why don't we tag this one out just to show?
Whoa. That's a big change.
Right, so now let's look at where our water level is compared to our lines. And you can see that water level is higher. And we can actually take the time to measure how much that water is raised up, and that tells us how much volume this takes up. And so for things like the fish and the things that call coral reefs a home, that change in volume, that amount of reef structure there is crucial. That's the big difference between having a home and not having one. So for these coral reefs to survive, we need to maximize the amount of volume that's happening in this 2-dimensional space, in this kind of finite square. We need to maximize the amount of volume that's happening in this limited space so that we can maximize the amount of homes for fish and lobster and all these other things that we care about on the reef, sea turtles.
I imagine if I were a little fish, like to be swimming around in here, staying safe, like hiding from bigger fish, maybe hiding from the work that I have to do. You never know. You know, that looks like a pretty good home.
Can you imagine like a big Nassau grouper trying to hide in there?
No, that would be very silly. A Nassau grouper would need a bigger coral, maybe one of those plating corals or a big sponge to hide in.
Right, so if a Nassau grouper is going to hide, it's not necessarily going to hide in this little coral, but then there's all kinds of little fish, little gobies, little cleaner shrimps and things that like to hide in this small space because they need that space for hiding, so it's an important place for their home. So let's see how best we can build our reef. So we've got our real reef here. Kitty, you want to switch with me?
Sure.
Right over here. So we've got our much bigger area that's our reef, right? It's still a limited area, right? We can still only put corals down in this rectangular space, but we want to maximize that change in volume that's happening here, right? Where this water line is, we want to get it as high as possible, right?
Right.
So let's start off first by marking our baseline. So when there's no corals, we have right there. And Sharpies don't work super great next to water, but
That's okay. We can see the mark.
I'm gonna put one over here because remember, like I said, we are tilted. So we've got our 2 baselines here. So let's start off by putting as many corals. So let's say we have a really efficient group, like these brain corals or the elkhorn and staghorn. Let's try and just put all of one type that's really fast at growing into the reef here. And so for that, I think we'll use these brain corals that we've used multiple times. So first, we've got this guy. That's gonna do some volume change. We've got
Good luck, Betty.
this guy.
Ooh.
Right, and remember, the corals can't grow on top of each other because they have to all be able to get some sunlight and see. So we'll try and put as many in here on that same ground as possible. But when we run out of space, that's the most number of corals we can use. So I think we're about out of space. What do you think?
Yep.
Right?
We had a little one, but for all the corals of about the same size and shape, I think we're maxed out.
Right. So right now we've gotten how many corals? Is that
1, 2, 3, 4. Oh, 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 oh.
was that your
That was my count.
your
reference.
count impression?
Yeah.
We always welcome the count, especially if he's a fan of Science Saturday. Most important. So let's make a mark of how much water we've displaced here. And you can see already, we definitely went up a good little bit.
Yeah, I'd say that's a solid centimeter.
Solid centimeter, says Kitty.
Yep.
Let's get a mark over here as well. While I'm doing this, is there any questions about any of the stuff that I've been saying? Any comments coming in, Kitty?
We're getting accolades. Oh, hey, Smushy Man. Smush is here. Great info. Thanks, guys.
Awesome. So let's try and rebuild our reef again. So let's take these guys out for right now. I'm gonna move the Sharpies. We'll see that it goes back down about. It's actually probably gonna go just a touch lower just because you notice that some of our corals are dripping.
Right.
So they're holding a little bit of water, so we're gonna lose a little bit of water between our trials, but it's not the biggest deal. Especially in a water volume this big, right?
Yeah, and imagine in the ocean.
Right.
But we're not taking corals out of the ocean, so it doesn't matter.
Right. So
We're
now we
saving
see
the corals.
we're back down to approximately where our original line is,
Mm-hmm.
right? And I mean we did go up a good ways. I would
Yeah,
say
that's a.
right. But
I
now,
think my measurement was off. That is more than a centimeter.
well, that's actually because of the properties of the water can sometimes magnify it, and so it's hard to tell
Ooh, yeah.
the actual difference. But now that it's both in air, you can see. So this is another point about what Kitty and I were just talking about. So this might be a really good coral, say for example, for like a Nassau grouper. Maybe it's a good place for it to kind of lay and rest and get its face and its scales and everything cleaned. It's a good like cleaning station for them. But it wouldn't be necessarily a good place for a little tiny like goby or something like that, right? And that's where it would be advantageous to have something like this on the reef too. So now let's try and rebuild the reef again, but let's try and get as many different kinds of corals that we can. So we'll start off, let's go ahead and get this guy in here. And he could probably, because he's so small, just kind of tuck away in that corner, right?
I hope I'm pointing at the corner.
What do you think, Kitty? What other corals do you want to add? We've got a few others here that we've used.
I know that it's a pretty old sample, but we do have an acropora here.
Right. Yeah, so here's an acropora, and you can tell like how big, and this is really thick. These are really fast growers. So we can kind of prop him up over there in that corner. Right?
Yeah.
And how about this flat brain coral, right? We're also going to put in here. But then also we have some here that are a little bit more bulbous. They're a little bit rounder, right? So let's get more of a round brain coral in here so we have a different shape too. What about this one? This might be too big.
That looks very bulbous. That might have to go right in the middle.
Probably does. And we have corals of all sizes on the reef, right? So we got some big corals and some little corals. Now, this guy, I think we would probably want to put him over there, but instead I think he can probably, because he's got this narrow piece here, he can just kind of squeeze in right there. Right?
So he's got his attachment point, but then his polyps go up and out looking for that sunlight.
Let's get, so I know we're working mostly with brain corals, but we'll work on some different shapes here too. So let's get this one over here. And it looks like we might have room for maybe one more if we can find the right colony. What do you think, Kitty?
I think that we can. And you know what? I bet that if that acropora were standing up instead of on its side, we would be able to get more in there too.
Oh, that's a good point. So let's pretend that the acroporas could properly mount,
Right.
right? Because we typically have acroporas grow kind of up. So we're just going to kind of move them up like that a little bit. So we still want them to be in the water.
Right. Maybe over here, going that way.
Oh, something like that.
Yeah. Wild.
Okay. So we still got this space.
No
over
big
here.
deal. I'm kind of an engineer.
I see.
I'm not at all. Sorry, guys.
I know a lot of engineers, so. So what about this guy? Let's see. I bet he can fit right in here nicely.
I would like to point out that we are officially above the previous water line.
Oh yeah, that's true. We don't really have any other coral shapes, unfortunately, that would fit probably pretty well in here, I don't think. Do you see any?
I bet if we moved the MMEA over to the corner by the Eusmelia, we could fit something in that corner there.
Okay, sure. Let's try it. Corals don't have this benefit of being able to get...
Right, no, this would not happen in the wild. Once you pick a space, you're there. And maybe that pointy one behind me could go up on its side.
Okay. Let's see.
Oh, this is nerve-wracking, guys.
Let's see.
I'm too short and can't see in there, so I don't know what I just suggested. Oh, man.
It's getting full, all right.
So now, how many coral do we have in here now?
Well, we want to make sure this is finalized here. There we go. There we go. We want to make sure everyone's nice and happy and submerged.
Yep.
So, yeah, how many corals? You got to do your count impression.
Okay. We've got, can we name the types too?
Sure.
Ooh,
Here we go.
okay, so in the corner we have our MMEA, which is a Meandrites.
Meandrina meandrites.
Meandrina
or the
meandrites,
maze coral.
or the maze coral. Then we have my favorite, the Eusimilia, because you smell like a flower. And 3, we have some sort of brain coral.
Pseudodiploria strigosa.
Pseudodiploria
The
strigosa.
symmetrical brain coral.
And then we got another one of those guys, so that's 4.
We might get in trouble with the count. I haven't cleared using his
Okay,
laugh.
yep. All right, we're going to pick another laugh. And then we've got our D-Lab and our Acropora.
Woohoo.
And I don't know what that is. Another brain coral?
Yeah, that's probably Pseudoploria clivosa. So it's a close
Ooh.
relative of these guys, but it's a little bit different. It
And
might
that's
also be a
7?
strigosa. It's hard to say.
And then number 8, another brain coral.
Right.
So we now have 8 corals in the bucket.
Right. And let's look at our measurement for our volume change. So we see our red line down there.
You're right, it's coral Tetris. This is fun.
Boop.
We do not recommend doing this with live corals or with corals that were not collected responsibly.
Yeah, we're doing this one so that you guys don't have to take the corals out. But when you guys are on the reef the next time you see and you're swimming around or you're diving or anything like that, you can look around, you can see how all these corals have evolved to tuck away in certain areas. So the Eusimilia here, well, it may not be as fast of a grower because it has such a small footprint, it can fit anywhere. And this is what Kitty was talking about, where she was like, it's really cool because you kind of find them as like little gems on the reef, right? Mm-hmm. And that's why, you know, when we're talking about like saving corals and things like that, it's important to save all of these species because they all contribute to this reefscape as a whole. And so now, compared to our first reef where we just kind of had the brain corals, there's so much more structure and places for different fish to hide and different animals to live. Not to mention, we've made a bigger, better, more diverse reef in the same amount of area, right? This is still the same rectangle.
Same bucket, same reef. And look at all those places to hide. So now the fish that was just hiding in our little Eusimilia, now he can go under and between and up by the Acropora and all around. And then you might have a big guy and he's like, I'm just hanging out over here because this is my coral head. And then somebody's over here like, I'm hiding over here.
Where'd you be hiding, Kitty? Where'd you go?
Where would I go? Actually, no, I'd probably go between the Eusimilia and the Meandrites.
Oh, really?
Like, in there, I'd be like, what? This is, like, my main house, but this is my other house because I'm a fish and I don't have to pay a mortgage.
You've got multiple... Real estate's all the rage right now.
Oh, yeah.
Especially on coral reefs.
Yes.
And interesting, I think you heard someone say it's like Tetris, right?
Yeah.
And so that also strengthens the reef, too, right? So if you can imagine if we had just kind of just those little bitty bulbs and we had less volume change, when you talk about things like storms and things like that and the wave breaking.
Oh, can we make a storm? It would be like, whoa. Oh, no, never
like
mind.
An earthquake.
That was an earthquake, and it was scary because I thought it was going to fall on me. So we're not going to do that again. But you guys can imagine that if something were to disrupt this area, this stronger, more elaborate reef would have more varied reef. Yes, would have more structure and a better chance of hanging on, rather than just one guy out there all by himself.
Right, or one guy and all of his clone friends, right?
Right. Yeah.
Right. So like, because of this, you're creating a much bigger reef in a shorter time, and that reef's going to be a lot stronger from storms. And you're creating more habitat.
Right.
And, you know, places where, say for example, because we could only fit 4 brain corals in here, right, now we can fit twice as many just because they're growing in different ways. And this is the reason that we see corals growing in different ways when we're on the reef.
Very interesting.
Right.
So, should we check and see where our water level is?
Oh, did we? Yeah, I thought we did.
Well, we marked it, but we haven't. If we take everything out, we can do another comparison.
Oh, to see how much water the corals absorb?
Yeah.
Yeah.
So while Joe's doing that, just because you just touched this, I just think that this is really cool. I found it on the ground. This is well.
Go ahead.
Well, I don't know. This is a cutting from a coral, but I think that it's really interesting because you can see that the shape and the structure, and it is just absolutely beautiful. And so you can see how the coral continued to grow up. And it just looks like a beautiful tile. I am not encouraging to use corals for tile.
No.
Just
But what you
showing
can
that
see
that
with this-
happened.
So this is basically like a cross section. You can imagine if you just kind of slice this like that. This is kind of what would come out. And so what you can see is basically, you know, we have this ridge and valley feature of the brain coral. And basically what is happening is just that coral is just building on top of itself, just kind of straight up. So this point here, this kind of little groove here, is just kind of slowly but surely, when the coral's alive, growing upward, adding a little bit more calcium carbonate, a little bit more stone underneath it every year, over and over and over.
Super cool.
And so that structure doesn't change through time. So this is just the inside of a ridge, and this is the inside of the valley. And so it just basically just creates another version right on top of itself through time. And that's how the corals actually build up and grow. That's what we were talking about earlier, where the coral's growing vertically in addition to horizontally.
All right, we're almost. Oh man,
Yes,
That's impressive.
all these little grooves and things, you know, they end up holding a lot of water as kind of a byproduct. So when we're actually doing this for scientific reasons, you have to be really careful that you don't spill a lot of water out of where this is, because otherwise you'll mess up your measurements.
right.
So you always have to check your baseline again. And you have to generally move a lot slower. I know when I'm in the lab and I'm doing either buoyant weights or displacement like this, you have to sit with your hand and let all the drips come off all the way.
Exhausting. We're not doing that today.
No.
We're doing fun science.
Hey, that's fun science too.
That's true. We're doing fast fun science.
There we go.
All right, so now we can see all of our marks here. So this is super cool. So that bottom mark, as Joe said, you know, the water level has changed a little bit from the corals taking it out, but look at the difference between the first with just the brain corals and then adding our dynamic reef added another centimeter. And though that's super cool. I think that's exciting.
Oh yeah, for sure.
All right, so I'm about ready for breakfast.
Yeah, that sounds pretty good. But is there any other questions about corals? Because you know I'm always happy to talk about corals.
That's true. Joe always likes to talk about coral. We don't have any more questions, although I really love the coral Tetris. Lucy, thank you. That was really funny.
Oh, there was something else I was going to show. So,
Oh.
so you remember when you tried to make your earthquake and that
Yes.
you were worried it was going to collapse on you?
Yeah.
You want to see something interesting? If we
Yes.
shake it just about the same amount, look how much more water is moving back
Yeah,
and forth.
a lot
constantly
more
like that.
movement.
So this is another form of how we can see that the corals act as that wave break. When you have all that tightly packed structure and that kind of it kind of cancels off all the energy that's in that wave. Because you know a wave, we see it on top of the water, but it's really going all the way down onto the ground, right? Especially once it gets close to shore. And so by having all that structure to kind of break up that wave motion, the more structure you have, the better it is at breaking up the wave motion. So
And
even-
that's why we have to protect our coral reefs, because they protect us.
exactly.
This is especially one of my favorite examples is coming into Christiansted, the barrier reef there. When you're flying in on the seaplane, it's very obvious.
Right. You can tell that difference between one side of the reef and the other, especially like when you're in that harbor area. That's the reason we're able, all those boats stay there, is because it's a safe haven. So, right. So it's a lot easier to generate this wave energy and for it to stay around and move uninterrupted than it is when we have the reef there. So, you, in a way, coral reefs are a habitat for a lot of things that we care about, like sea turtles and fish and lobster and conch and things, but it's also a habitat for us, indirectly, because it creates the area that we live a place where we can continue to live. And that's why it's so important to save, and not just one kind of coral, but as many kinds as we possibly can.
Absolutely. All right. Well, guys, I think we're coming up on the end. I want to thank all of you who've come in, and we're going to give an extra round of applause to our favorite coral scientist, Joe, today, because it's his birthday, and now we're going diving. So, everyone, thank you. Have a wonderful day. Back on me. Thanks again. It's always great to see you guys, and new names in the attendance list. So welcome, guys. Thanks for coming, and we'll see you next month. Bye.